And if you guys haven't already, go ahead and log into your SMCE account. Put that link in the chat one more time and make sure that you're selecting the small server on demand. And as everyone's waiting for that to load, again, interrupt me if you guys have any questions from yesterday's material. And just a reminder that I do update the schedule at the end of every day based on what we got through. And so you guys take a look at the current schedule in the link that I just put in the chat on GitHub. And so yesterday we went through and submitted our jobs to assess strandedness using the RCQC Infer Experiment module. And today we're going to compile those results from all samples and then answer some questions about the strandedness of our RNA-seq data. And then we're going to hop back into the lecture and discuss how we quantitate those data. And then we're going to jump back into the Jupyter Notebook and actually quantitate our data, creating our final counts table. And then we'll get into a break. And after our morning break, we'll hopefully start our debrief section on the RNA-seq overview through the counts table, as well as the FASTQ to Counts Jupyter Notebook. And that'll bring us into lunch, and then we may need to carry that over after lunch. And we'll also be finishing the RNA-seq overview lecture after lunch. And then we'll start getting into our next Jupyter Notebook, which is beginning to run differential gene expression using the DE-seq2 algorithm. If any of you guys have a screen like this, you can either click keep waiting, or it might just load like mine did. And once you guys have logged on, make sure that you're seeing, I'm just putting the link to the SMC in the chat again for those of you who just joined. Go ahead and log on. Small server on demand is the one that we're selecting. And once you're logged on, make sure that you're seeing down here on the left that you're running the Python kernel, conda environment :GL4U, and it should say idle. If anyone doesn't see that in their bottom left, let me know. And how are you guys doing with your instances? Is there anyone still waiting? Okay, I'll give it a bit more time. While we're waiting, again, if there's any questions about anything we've gone over, let me know. You can unmute your mic, type it in the chat. Anything you want to hear explained again. So to bring everyone up to speed on where we are in the workflow. So we started with our raw reads in FASTQ file format. We assessed the quality of our raw reads. We noticed that there were some lower qualities towards the end of those reads, particularly in the reverse read. We also noticed that there were some adapters that were in our reads. And so we went ahead and trimmed and filtered our data using the tool TrimGalore, which uses CutAdapt, another tool underneath the hood. After we finished trimming and filtering, cleaning up our raw data, we then did another QC assessment on our trim data, and we noticed that we got increased quality, especially towards the end of those reads, because that's where TrimGalore trims from. And then we also noticed that we successfully removed all of our adapters. So we felt confident then that we were able to align those reads to the reference genome. And so we did that using a tool called STAR, and STAR needed to format our reference. I do not have the Python idle on the bottom left. Should I restart? Yeah, if you guys aren't seeing this Python conda environment GL4U idle, you can go up here to the top right, or apparently you can click down there too. And then in the drop-down menu, you want to select Python conda environment GL4U, and then click select, and then you should see down here in the bottom left that it'll initialize, it'll connect, and then it ultimately should end up at idle. So Jen, if you're not seeing that, let me know once you restart your kernel, if you're still not seeing that. Okay, then carrying on, the STAR alignment tool needs our reference index or a reference index, so into a specific format to allow STAR to easily identify regions of interest, so our annotated genes, when it's going through and doing the alignment. And if you guys remember, at GeneLab, we use references from the Ensemble database, and since we're working with samples derived from the soleus muscle of mice that were flown in space on the International Space Station, as well as respective ground control animals that were kept on the ground, we use the mus musculus reference. And then I showed you that in order to create an index with STAR, we only need to do that one time per read length, because there is a segment where we indicate the overhang that we want. And of course, we want that overhang to be up to our max read length -1, that overhang being how it identifies splice annotations. And so spliced regions, so it could be, that read could be spliced all the way up to just having one base pair on another exon from the read that you have. And so we were all able to use the same STAR index, and then we went ahead and aligned our reads with STAR. We took a look at those BAM files. We went through and looked at what all those columns mean. You guys answered some questions about the information provided in those BAM files. We noticed in the BAM file that we were looking at that we had 2 mate pairs in our data shown in the first 10 lines, and one of those mate pairs mapped to 2 different transcripts, and the other mate pair mapped to 3 different transcripts. So both the pairs that we saw in the beginning of that file for our flight rep 1 sample had multi-mapped reads. We then took a look at the log file, the log final out file that STAR generates, which is a summary of how the mapping went. And then we compiled all those log final out files from all 12 of our samples using multi-QC, and then you guys answered some questions on how well the mapping was, and we saw that all of our samples had over 75% of their reads uniquely mapped, which is great. We then went ahead and indexed those alignment BAM files, so that the indexing of those alignment files is required to assess strandedness with the RCQC infer experiment module. And we went over in the lecture that this RCQC infer experiment module needs to know the annotated genes, but wants those annotated gene information, so the genomic coordinates of the genes in our reference, to be in a certain format. So we essentially played around with these 2 tools, GTF to GeneFred and then GeneFred to BED, to ultimately go from a GTF file that we pulled from Ensemble to a BED file, which is all of the genomic coordinates for all of the annotated genes in the format that's expected by this RCQC infer experiment tool. And then we went ahead and went over the options for the infer experiment, and everyone ran that tool just before we left yesterday. So we're going to pick up right here in 6A, and we're going to start taking a look at that strandedness data. Is there anyone that's still waiting on their instances to be up? Is there anyone not seeing the Python const environment GL4U idle in the bottom left of their hub? Okay, great. And then just as a reminder, as you guys are toggling back and forth while answering the questions, if you go to the left-hand panel, on your screen here, you'll notice everyone's probably, unless you changed it yesterday, still in the file browser mode here, which is looking at all the files in our home directory. So if you guys want to see the table of contents, you can just scroll down to the bulleted list icon just above the puzzle icon, and it'll pull up the table of contents, and you can then hop around to different places in the notebook using that. And so we're going to hop to 6A, which is the evaluate strandedness section that we left off with yesterday. And so we went through, I just gave you a quick reminder here that in addition to infer experiment, we also use this RCQC tool suite to do some additional analyses and assessment of our alignment data. So these are the 3 additional RCQC tools that we use. So if you're navigating the GeneLab website for processed data, you might see some of the outputs from those analyses as well. So I just wanted to have the links here in case you guys are curious and wanted to learn more about them. Of course, we don't need those for the processing pipeline. It's just another way to assess the quality of the data. So before we left yesterday, we went over the infer experiment, which required that reference file embed format that we had made previously. It also required our input BAM file, which they're expecting the aligned to genome BAM file. Keep in mind that star, because of the options that we put, outputted 2 BAM files, one with alignments based on genomic coordinates and another with alignments based on transcripts. And so we're using the genomic coordinate one. And then this S is how many reads we want to sample to assess the strandedness. And as I mentioned to you guys, at GeneLab, we played around with different values here and ultimately settled on 15 million reads was sufficient for us to sample in our data to be confident in the strandedness of our data. And then this tool will output its results in standard out. And so when that happens, I do like to redirect it to its own file. And so that's why I add this little caret here and then redirect that output to our directory structure that we set up way at the beginning. And then have that name be the sample name and then just the name of the analysis it was run, which is infer experiment, and then .out, because it was the output of that tool. And so that means our input data is going to be that bed reference file that we generated above from the GTF file, and then our genomic aligned BAM file from each sample. And our output data will be that infer experiment.out file again for each sample. So when we left off yesterday, we had submitted our Slurm array script to be able to run this same command on all 12 of our samples. And then we opened up what one of those output files looks like. So if you guys haven't done this at the end of yesterday, go ahead and run this code block to cat or open up the flight rep 1. So this is the first biological replicate of our flight samples. And it's the infer experiment out file. And this is what you guys should all see. Is there anyone who's not seeing this? Okay, great. And here we did take a quick look at it just at the end of yesterday. And so I go through and I break this down similar to how I did in the lecture of what this information needs, or what this information means for single end data and what it means for paired end data. So because we have paired end data, we'll focus on that. And so essentially the fraction of reads explained by this being the forward read, anytime the reference is in the positive direction, the read is in the positive direction. Anytime the reference is in the negative direction or antisense direction, the forward read is in the antisense direction. And then it's always going to be the opposite for the mate pair. So in this case now the reverse read, anytime the reference is in the positive or sense direction, the reverse read would be in the antisense direction. Anytime the reference is in the negative or antisense direction, the reverse read will be in the positive or sense direction. And we just note here that for this sample, we see a very small fraction of the reads explained by that. And so then we also have here the reads explained by this, and this being that for the forward read, anytime the reference is in the positive or sense direction, that forward read is in the opposite or antisense direction. Anytime the reference is in the antisense or negative direction, that forward read is in the positive or sense direction. And then again, always the opposite for the reverse read. And so in this case, the reverse read, anytime the reference is in the positive direction, the reverse read is also in the positive direction. Anytime the reference is in the negative direction, the reverse read is also in the negative direction. And we see here that the majority of the fraction of sampled reads is explained by that particular orientation. Again, always with respect to the reference. And so I want you guys to take a look at these data for this one sample and answer these questions. So Lisa, if you could pull up this question on the Mentimeter link, and I'll give you guys a minute or so to answer what percent of reads have undetermined strandedness. And if no instructors answer, we should have, I believe, 9 answers in total. Yeah. So Lisa, let me know we're getting close to that 9 number. So go ahead and answer the question, what percent of reads based on this output for this sample have undetermined strandedness? Go ahead and put in your answers or guesses. I'm going to show. $13.4, 13.4, fraction, 0.134, 30.4. Okay, most of you guys got this right. Yeah, so it's right here at the top. So there's usually going to be some percentage or some fraction of the read sample that failed to determine strandedness. Again, this could just be because those reads might be palindromic, meaning that they read the same forward and the reverse orientation, so it could be difficult or impossible to know what the strandedness actually is, especially if they're smaller read fragments. So yeah, a lot of you guys got that right. This information, again, right up here at the top of the output for infer experiment, it'll tell you what the fraction is. And then, of course, just multiply by 100 to get the percent. Great job. Let's go ahead, Lisa, and pull up question number 2, which is what percent of reads are sense relative to the reference? So what percent of reads are sense, meaning in the same direction relative to the reference? And I shouldn't say reads here, I should say insert, because I know we have forward and reverse reads. So I mean the insert. So this would be the forward reads. What percent have forward reads in the sense direction relative to the reference? Which ultimately means the insert or template that was sequenced is sense relative to the reference. I'm going to go ahead and show the results. 0.76, 0.76, 0.76. Yeah, a lot of you got those right. One not sure, so let's go over this. So we have the percent, again, of forward reads, I'm sorry, I'll correct this, or entire template length that's sense relative to the reference. So we're looking at the fraction of reads explained by this orientation. And so this being that the forward read, for the forward read, anytime this first plus sign, that's referring to the template, because keep in mind, let me just go back up here, keep in mind that our template, the orientation of our template or our reference could be sense or anti-sense. Again, it depends on how that reference was created. So we do have some of our references that are in the sense direction, as you see here in this example. We see some of our reference transcripts are in the anti-sense direction, as you see here. So we're always doing relative to the reference. And so taking a look back at the notebook, that output for infer experiment is for paired N. It's going to tell you first information about the forward read. So this is saying anytime that reference template is in the positive or sense direction, the forward read is also in the positive or sense direction. And then anytime the reference is in the minus or anti-sense direction, the forward read is also in the minus or anti-sense direction. So in this orientation, the forward read is always in the same orientation of the reference. And then the reverse read, of course, because you're reading the opposite direction, so the reverse read should always be opposite of that. So that means that in this orientation for the reverse read, anytime the reference is in the positive or sense direction, the reverse read is going to be in the negative or anti-sense direction, again, opposite the reference. And then similarly, anytime the reference is in the minus or anti-sense direction, the reverse read will be in the positive or sense direction. So that means that your entire template would be in the sense or same direction as a reference, so similar to this picture here. So we have this reference in black in the 5' to 3' direction, And then we have our forward read also. When it lines up to the template, it lines up in the 5' to 3' direction. So again, same orientation. And the reverse read would be opposite of that. So this would be the 5' end over here, and this would be the 3' end over here of the reverse read. So opposite orientation of the reference. So that means that that template is going to be in the same, the template that was sequenced is going to be in the same orientation relative to the reference. And so the fraction explained by that is, as many of you guys got it, fraction is 0.0076, multiply that by 100 to get the percent. Is that still unclear to anyone? Okay. Awesome. Yeah, it's okay it took me a long time to wrap my head around this, so this is new to all of you. So if this is a little confusing, I'll do the best I can to explain better or have one of the other TAs give it a try. So that's the fraction of the forward reads or the template that's in the same orientation relative to the reference. So our next question, Lisa, if you want to load it up, should be what percent of the reads are antisense relative to the reference? And again, this should be what percent of forward reads are antisense relative to the reference, or it could say what percent of the templates are antisense relative to the reference. So I'm sorry for the confusion there. I should have been more clear because this is paired end data. So what percent of forward reads or entire templates are antisense relative to the reference? Okay, let's take a look. Well, 85.79 across the board. Maybe one person that isn't sure. Okay, great. I'm rounded. That's a problem. Yeah, so to find out what percent of reads are antisense, again, forward reads, because this is paired end data, I will change that. What we want to look at is this down here. So this is going to be the fraction of reads explained by this orientation. And again, this is opposite the orientation explained above. So this is anytime for the forward reads, anytime the reference is in the positive or sense direction, the forward read will be in the anti-sense direction or negative direction. Anytime that reference is in the minus or anti-sense direction, the forward read will be in the positive or sense direction. And then again, always opposite for the mate pair. So for the reverse read, anytime the template is in the positive direction, the reverse read is also in the positive direction. Anytime the template is in the negative or anti-sense direction, the reverse read is also in the negative or anti-sense direction. And so the fraction explained by this is 0.8579. Multiply that by 100 to get the percent of reads. And so now that we know the fraction or percent of reads explained by these two different orientations, either sense relative to the reference or anti-sense relative to the reference, Lisa, if you could cue up the last question in this section, which are these data stranded?
We have 9.
Awesome. Let's go ahead and do those results. Yes, anti-sense, all yes, some with some question marks, like on a deserted island. I don't know what that means. Okay. So yeah, they are stranded. So most of you guys got that correct. And the person that put anti-sense, that is true. So if these were unstranded, we wouldn't see too much of a difference between the templates that were in the same orientation relative to the reference and those that were in the opposite orientation relative to the reference. So if these were unstranded, thanks, Mike, for the clarification. So if these were unstranded, we wouldn't see too much difference between the orientation of the reads relative to the reference. So we would see about half the reads that were in the same orientation and the other half of the reads that were in the opposite orientation. So we would see both of these lingering around 0.5. However, if we see that a strong or large portion or percent is in one orientation with respect to the reference than the other, that means that they are indeed stranded. So we are seeing stranded data here, and that makes sense because the kit we used from Illumina, as we went through during library prep, is indeed stranded. And we notice that it's stranded in a specific orientation, and that orientation is going to be antisense relative to the reference because we see the largest fraction of the reads are in the opposite orientation relative to the reference. So these are stranded data, and they are in the antisense direction, again, relative to the reference. Is there anyone where that's still not clear on? Of why these are stranded and why they're stranded in the opposite direction relative to the reference? Okay.
Excuse me, sorry. Can you go over that just one more time? I'm having a bit of an issue just wrapping my head around it.
Yeah, for sure. So let me see. Is this why I hate not having a whiteboard? But I'm just going to go back to this example here. So here we have our reference, and let's say that this reference represents one transcript. So let me get rid of these. So let's say that this is our transcript, right, from the 5' to 3' direction in our reference. So 5' to, our reference in this example is in the 5' to 3' direction, so that is the sense or causative direction. And then when we went ahead and, I'm going to move these back to where they originally were. When we went ahead and mapped our forward and our reverse reads, what we noticed is that the reverse or the forward reads mapped in the same orientation relative to the reference. So these arrows indicate that the side of the arrow is 5' and the side of the arrow is 3'. And then, so anytime you see the head of the arrow, it's considered 3'. Sorry, 5' again, because you always, a sense is always 5' to 3' direction. And so in this example here, where we have the template that's in the 5' to 3' or the sense direction, when these reads were mapped, they were mapped such that the forward read mapped where the 5' end was mapping to the far left and the 3' end was mapping more to the right relative to the reference. So it was mapping in the 5' to 3' direction, the same direction as the template, which is also 5' to 3'. And then the reverse read, again, because it's always going to be sequencing in the opposite direction of your template. So then in this case, the reverse read, when it was doing the alignment and going to map to the reference genome, what happened is in order for that read to map successfully to this transcript, it had to be flipped. So the reverse complement had to be looked at such that in order for it to map successfully to the reference, it mapped so long as it was in the opposite orientation of the reference. So when this matched up to the reference, it was the 3' end of that reverse read that was to the left of the reference, and the 5' end that was more to the right or to the 3' end of the reference. So in this case, we have our forward read in the same orientation of our reference, and the reverse read maps in the opposite orientation relative to the reference. So that means that the template that was sequenced, because we know it was sequenced in one direction, in forward direction, and then in the reverse direction, so this template must be set up and must map in the same or sense direction relative to the reference. So if this is the case for most of the reads, where it's in the same orientation relative to the reference, it would be considered sense or in the same direction relative to the reference. However, it could happen, and does happen, where let's use the same example of this template or reference this reference being in the 5' to 3' direction. And then when you go to map the reads down, this is actually, change the orientation. So this could be such that it maps like this over here. And then this now, in this example, hard to move things around here. So that would be if it was stranded in the same, the one that I just went over would be if it was stranded in the same orientation. But it could also be the case where it's in the reverse orientation here. So in this case, put it on the map up there. Oops. Sorry. Push it over there. And we run. All right. The 3' should be on the other side. Don't know why I can't just move it alone. So in this instance, what we have here is that the reverse or the forward read in this example is lining up such that the 3' end maps more to the 5' side of the reference, and the 5' end maps to the 3' side of the reference for the forward read. And the reverse read, again, always going to be opposite. So in the reverse read, we have it mapping to in the same orientation as the reference. So the 5' end of the reverse read is more on the 5' side of the reference, and the 3' end is then also on the 3' side of the reference, and it maps like that. So in this case, what you would have is your template, since your template is reading from forward to reverse, would be mapping then in the 3' to 5' orientation relative to your reference. So in this context, which is what we see in our data, we do see that we have data that are stranded because the bulk of our reads are mapping to the opposite orientation of the reference, but it's antisense because it's mapping in the opposite or reverse complement orientation relative to the reference. So it's still stranded because we still have the majority of the reads or templates in one direction relative to the reference. So that's how we know it's stranded. If the majority of the reads are mapping to one direction relative to the reference, that means stranded. And then you know if it's sense or antisense, depending on if the majority of the reads are mapping in the same direction as the reference, or if the majority of the reads are mapping in the opposite direction of the reference. So if the majority of the reads are mapping in the same direction of the reference, it's sense relative to the reference. If the majority of the reads are mapping in the opposite direction relative to the reference, like we see here, it's going to be antisense relative to the reference.
One quick question. What does the green represent here?
That's the template. So the template that was sequenced, so that's equivalent to, if we go back up to our library prep.
I see. Thank you very much.
Have these clusters, right? And so we're sequencing it in one direction and then the other. So that template represents the green area here. So the part that we're sequencing that came from our sample.
Right, thank you.
Template, right, is going to be from the start and end of the forward and reverse read. And the size of this template depends on where those reads align to.
So Amanda, can you go back to the first strand synthesis just to review? Because essentially, I just want to be sure. When you do stranded libraries, you eliminate the second strand cDNA because to make it stranded, is that right?
It depends
By
on
incorporating uracil?
That's how this kit works, yes.
Can we look at that again? I just want to combine these two ideas.
Yeah, so different kits will work in different ways. For the Illumina strandedness kit, what they do is they synthesize their first strand of cDNA from the RNA template, And then when they synthesize their second strand of cDNA, they incorporate uracil instead of thymine, so that when you go through the last step of the library preparation, which is amplification, it uses a polymerase that can't go past a uracil, so only one orientation of the strand is going to be amplified. And
But
so
if you go back to the previous slide,
this one here or this one here?
yeah. So it was made 5' to 3' on the first strand, but the beginning of the gene and the template strand is on the 3' end. Sorry, on the 5' end. So if you think about it, this was the reverse primer for the first strand, correct?
Yes. Well,
So the first strand is preserved. The first strand is made with the reverse primer. The second strand is made with the forward primer. But they just always show it in the direction of synthesis, which is always 5' to 3', as opposed
Right.
to the transcript. So you have to combine those. And so you eliminate the second strand and you're always amplifying the first strand,
right.
which was made with the reverse primer. Yes?
Yes.
Okay.
Yeah. Yeah, and if you follow suit, and the kits tell you what strandedness it should be at the end relative to the reference, but again, it's not always clearly indicated in the documents, and then you can mix and match. So what happened with us is you can mix and match your adapters. So we get our adapters from IDT, and our IDT adapters actually reverse the priming sequence on the two sides, and so it ends up being the reverse orientation of what the Illumina kit tells us it is, because we're getting our primers not from Illumina, our adapters not from Illumina, but from IDT. And so, yeah, we ran into that.
It would be nice to prepare a slide deck that always had the template transcript, sense, antisense, coding, non-coding. That language can be confusing. Template strand, which is a transcript, 5' to 3', and kept that in the same orientation and went all the way through and showed what is happening, because things get flipped. And so you're
Yeah.
doing this logic, negative of the negative of the negative, at least in my mind. And it would help to just make one slide. For example, that first strand cDNA, the green should have always been on the, for the convention's sake. But anyway, I think that would be helpful for me in teaching, so that I can have a consistent flow for students to show, start with the transcript and here's what's happening. But
Okay.
remember, you started with this transcript and this orientation.
I can work on that. It gets complicated because remember, when it sequences, it actually makes a copy of it, and then it's the
Yeah.
copy that's ultimately sequenced, because then it's tethered to the flow cell. But yeah, I could work on a slide like that.
But I think that's why, yeah, anyway, it would just help to follow, because there's the chemistry of what's happening. And then it's important to always go back to that when you're trying to understand the language of reverse and forward and read and this and that, which is,
Yeah, and then it's also confusing.
relative to which frame of reference.
It's always
is it
relative.
forward or reverse?
Yeah, exactly. 'Cause that's
So,
all
but there's an
confusing.
ultimate frame, and that's the 5', 3' sequence of the transcript.
Yeah, I could work
That's
on.
ultimately what we started out with.
Yeah, I could work on a slide like that, so that you guys can have it when you teach at your home institution. And
Maybe
then
we should work on it to make sure that we understand what's going on, and then you can
and
see
then
if they're
I
correct.
can review it. Yeah, sure. That would be take work off of me. But yeah, I'll be happy to work on one as well. And then if you guys want to work on one yourselves, I think that would be great, because exactly that'll help with your understanding of it too. And then let's see, why Jen?
Oh, sorry. I had a question also on library prep.
Mm-hmm.
So if our mRNA or a fragment of the mRNA is GC rich, this chain termination method for library prep doesn't work anymore. So when we go on and determine strandedness, are the values overrepresented? Because we can't
I get what
tell
you're
anymore.
saying. So you're saying that if you have a GC rich region, it might be a while when you're doing your amplification before your polymerase is able, yeah. So how that would come up is if there are pieces, or even if, you know, there's like, maybe there's no use here at all in one or more of your templates that you're sequencing, that would come up in the fraction for the strandedness. So if that's the case, then you'll have some templates, and that happens, that are unstranded, meaning that you see it mapped to the template in both the sense orientation and the anti-sense orientation. So how that would show up, and you're right, and sometimes that happens, or sometimes there might be issues with how it's preserved or what strands are selected in the sequencing that could happen, and how that would show up basically. And what you noticed here is that you never get fully stranded, right? So there's always some fraction that's the opposite orientation. So even though we have, in this example here for paired end data, the majority even more so than ours actually, but even if we look at ours, right, we only have, again, we sampled 15 million reads of this sample, and we only have 85.79% that were in the opposite orientation relative to the reference. So 85% is a decent amount, enough for us to be confident to consider this stranded. But you do notice that we do also have a small fraction that is sense relative to the reference, right? And so there must be some templates where we're seeing both, and that could be the case, that maybe it's a template that's GC rich and there was just no uracil there, and so both the first strand and the second strand in the library prep were amplified. And so there could be some of your templates that you can't determine the strandedness. And so that's why you're always going to see some fraction, even in stranded data, where you have both strands too. Yeah, so that's a good question, good point. And again, it shows in the data here. And then YGen, to your question, is there a cutoff percent to claim stranded? No, there's no like set cutoff here. At GeneLab, we have a 65% that we go with. Again, that's just looking at all the RNA-seq data we've done so far, and knowing information, when we know information about if the library prep kit was stranded or not, taking a look at the data. And if at least 65% is in one direction, we say, yeah, that's probably stranded. If we do see that's like hovering right around 65 or below 65 in one direction, that means that it could be unstranded, or maybe we just, an issue happened like the one that was just brought up, and for whatever reason, we couldn't determine strandedness from our data. So in that case, we could also go back and sample more reads. So again, we sampled 15 million reads. So maybe if we took a look at even more reads, we would see a stronger lenience toward it being stranded or unstranded. And then of course, if you know if the kit is stranded or unstranded, then that can help you make a final decision too. But yeah, there is no hard and fast rule of if it's always going to be this percent or higher if it's stranded or not. It depends on how good your library prep went, and on the data and the contents that you're sequencing. So good questions. Yeah, no problem. Anyone have any other questions about strandedness? It is a lot. It's a lot to wrap your head around. We're making copies of things, and we're making copies in order to sequence, and then in sequencing, you're actually synthesizing a new copy. So every time you're making a copy, right, you're generating the reverse complement of the strand that you're working with. So it's a lot to wrap your head around. To follow it can be tricky. Okay. And then just like for all of our other analyses, right, we ran this on every single sample in our study. And so rather than having to go through and look at the output for every sample, for us, we have 12, so it probably wouldn't be too bad. But when you get, 50, hundreds of samples in a study, it could be really cumbersome to check to make sure every sample is either stranded or not stranded. If it is stranded, if they're all stranded in the same orientation. And so we get to turn to our lovely tool multi-QC again, which can also read the output for the RCQC and for experiment data. And so we can use multi-QC again to compile all of the infer-experiment strandedness information from all 12 of our samples. So that's what we're going to do now. And so again, you guys are probably very familiar with this command now. Call it with multi-QC. -n is the prefix name for the output files. This time we're going to do infer experiment multi-QC. Always keeping in mind when you name things, you never want to use spaces or weird characters. So that's why I use underscores to separate information in the name. -O is the output. So this is going to be generated in that nice directory structure that we set up way at the beginning under the RCQC analysis and for experiment in its own subdirectory for the multi-QC report. And then as a positional argument is a directory holding all of the outputs for each sample for infer experiment. And so if you haven't done so already, you can go ahead and run that. Mine's taking a little bit. And you notice here as it's running, just keep in mind, you know, things are running because you should see a star in the brackets next to the code block. And then also down here on the left, you'll notice that it's telling me that my kernel's busy. So that means that it's running something. So I'll give this a little bit more time. Is there anyone else who's still waiting like I am? Yeah, still waiting. Okay. It's not just me then. So we'll give this a little bit of time. Okay, so Susie mentioned that hers was having some trouble too, and she ended up having to refresh. So these things, as you guys remember from previous, usually run pretty quick. So if you're still waiting, maybe do the same thing with me. You can go ahead and press stop and see if it'll stop it. Make sure that it says idle down here, and then you can try to rerun it again. Oh, I know what's happening, guys. I am so sorry. Let's stop this. I did not run the variable command at the top. Yeah, really sorry about that, guys. So it's probably trying to look for a path that doesn't exist. So if we can all scroll back up to the top and then run this variable command block under define paths. So go ahead and run that block. It should populate with a new number once it's complete. And once you've run this code block, again, this is just redefining the variables that hold the paths to where all the data are that we're working with. And then you can hop back down. You could go to your table of contents on the left if you have it pulled up, hop back down to step 6B, and then go ahead and try rerunning that. Sorry about that. That looks better. So hopefully that solved everyone's issues. Thank you, Susie, for pointing that out. Is there anyone who's still waiting? Awesome. Everyone was able to run now that they did the variables. Yeah, sorry about that. And so awesome. So this looks like it finished completely. I know we went over this a bunch of times. So it tells you the version. It tells you if there's a new version available. It tells you the path that it's looking in. It'll find anything that looks close to an output file for any of the tools that multi-QC works with. And so what we're looking for, because these are RCQC, a tool that we use. So we see here that it found 12 infer experiment reports, which we expect because we have 12 samples. And then just some information about compressing the data and then putting it out. And then we always want to make sure it says multi-QC complete. We don't see any error warning messages, so it looks good. So if anyone's seeing anything different than what I have here, please let me know. And then just like all the commands on here, we have parameter definitions written out. And the input data, of course, is our inferexperiment.out file, so our output from inferexperiment for each of our 12 samples. And then the output data, as we're all used to now from multi-QC, is going to be a directory containing all the information from, in this case, the RCQC inferexperiment data, and then an HTML file that puts that information in a nice graphs. And so we're going to take a look at that HTML file now. So we always want to import the display iframe function of Python. So go ahead and run that block if you haven't already. And then we can view the multi-QC report, this time for the inferexperiment command that we ran. And again, multi-QC, once you're inside of where it's being displayed, you can scroll left to right or up and down. If you ever want to change the display, you can just modify the width and the height of the display that you're seeing. And so if we scroll down here, this is going to be a pretty simple report because there's not a lot of information in inferexperiment. It's just telling us the strandedness of our data. And so what you'll see is something that looks like this. So again, each of our samples are listed here on the left. Down here on the bottom is the percent of total reads for each sample. And then the amount of samples that are in the sense or the same direction relative to the reference are in light blue. So you see for most of these, very little. And then the percent of total reads in the anti-sense direction relative to the reference is going to be in black. And the percent of reads where strandedness could not be determined is going to be in green, which you see here on the end. And then if you want exact values of any of these, you can always just hover over and it'll tell you the exact percentages for each of your samples. So this is really nice because it's very clear from looking at this graphically for all of our samples what the overwhelming orientation is for all of our samples for the templates that we've sequenced relative to the reference again. And so I want you guys to take a look at that. And then we have 2 questions. So taking a look at that multi-QC report of the strandedness above, Lisa, if you could queue up this question here. Are the data for all samples stranded or unstranded? You guys can go ahead and write that answer in the Mentimeter link. And Lisa, let me know when we have responses from everyone.
I believe 8 so far, just one more.
Awesome. Give people a few more seconds.
We have 9.
Okay, wonderful. Let's take a look. You guys said, are the data for all samples stranded or unstranded? All of them are saying stranded. Yeah, some of you guys are saying the majority are stranded. So I would argue that all of the samples are stranded. So for those of you guys saying the majority of the samples are stranded, are you referring to the majority of templates in the samples are stranded? Because it looks to me like every single sample is overwhelmingly stranded in one direction, but of course every sample does have some percentage or fraction that is the opposite of what the majority is in terms of strandedness. So yeah, here we see overwhelming, the large percent, well over 65%, closer to 85, 90% of the reads, ultimately templates for each of these samples are anti-sense relative to the reference. I gave the answer away to the next one. So they are sense, or are stranded data for all samples. And then the last question, I'll give you guys the answer to that. So if stranded, what is the orientation of the reads relative to the reference? Give you guys a few seconds to answer that. Lisa, if you could pull it up. How we doing on the answers, Lisa?
It's 7. 8.
Answers. So if stranded, what is the orientation of the reads relative to the reference?
We have all.
Got them all?
Mhm.
Awesome. Yeah, antisense, yes. We got one forward, so let's take a look here. So we see it actually, the nice part about multi-QC is it tells you with these color coding things what direction they are relative to the reference. So we see that if this was majority light blue, then it would be sense relative to the reference. What we're seeing is that the majority of the templates for each of the sample are in the anti-sense direction relative to the reference. So it's nice because it actually tells you the orientation relative to the reference in terms of these color coded, and then there's some fraction, some percentage that's undetermined where the strandedness couldn't be determined. So for the person that said forward, is this clear? If this still isn't clear, please let me know in the chat. Okay. And then I'll work.
Amanda, quick question. So at the informatics level, then you do not need to take the reverse complement, but the program does it anyway, right?
When it goes to map down the reads,
Yeah.
it'll see
Yeah.
how the reads map, and the reads can map either, in the orientation that it is, or it might need to be reversed in order to find a map. Yeah, so it'll look.
So in this instance, because we did the first cDNA, which is going in the other direction, right? So it doesn't need to take the reverse complement, correct?
Well, it'll look at that. So I'll put
It'll look
together.
at that, but it's aligning using the sequence as is because it's the first cDNA strand, which already was in the other orientation.
For the reverse reads.
Yeah.
So the reverse reads, the majority of them are going to align in the same orientation as the reference. For the forward reads, when they went to go try aligning those forward reads, they had to flip
Yeah.
them. And so because they weren't going to match in the
Right.
direction 5' to 3' the forward read was, it had to be the reverse complement 3' to 5'.
Exactly.
in
So,
order.
okay, got it. So the reverse read maps as is, the forward read have to reverse complement to
In order
map.
to map. Right.
Okay.
Right.
Okay.
I know.
I'm appreciating microarrays right now.
Oh, you get so much more information though from.
Yeah, but I'm not sure if I believe this information where you're like, we haven't even gotten to the algorithm for deciding whether it's a match or not, and you're reading, I don't know.
But you could always do it
Not
array.
an experiment. This is the beginning of an experiment. It has to be confirmed through other methods anyway, so, but
It is always good, to verify your data using an additional method for sure. Okay, so now we officially assess the strandedness of all of our samples. So we know that our templates are anti-sense relative to the reference. And so now we know when we quantitate them, we just want to quantitate the templates that are aligning to the anti-sense direction relative to the reference. So we're going to hop back into the lecture and go over how to quantitate these data. So just to bring us back to our workflow here, so we've evaluated our raw reads, we've trimmed and filtered them, and then took a look, made sure they were good and ready to go to be aligned, created a star index to be able to use the star aligner to align our reads to the reference genome, took a look at the mapping quality, saw that we're confident in the mapping quality with over 75% reads uniquely mapped for all samples. We then looked at the strandedness to be sure of what the strandedness of our templates are that we sequenced, and we now know once we compile that information with multi-QC for all samples that all of our samples are indeed stranded, and they're all stranded in the anti-sense direction relative to the reference. And so now we are finally at the step where we can start counting and getting some expression level data. So we're going to use a tool called RSEM to do our quantitation. And RSEM, similar to STAR, needs to create an index of the reference that we're using in order to do the quantitation. And so let's get into quantitation a little bit more. So of course, to determine the expression level of each gene in each sample, we next have to assign or count the align reads. This is where we have to assign it to a transcript and then count how many of our templates are from that particular transcript. And so there are several tools available, just like all other steps of this pipeline, there are several tools available to quantitate gene expression data, some of which are also aligners. So there are some aligners that will spit out quantitation data if you put the option. STAR is actually one of them. And they're going to use different algorithms to do the quantitation. And so some of the main differences between these algorithms are whether they're non-weighted or weighted for the quantitation. And essentially that'll have to deal with how they handle the multi-mapped reads, or reads that map to multiple genes or multiple splice isoforms of the same genes. And that they're going to differ in how they output their quantitation data too. So just as a reminder here, multi-mapped reads, this is looking at an example of a read that'll map to more than one gene. And so this is where we have the instance where we have this read here in purple, and it happens to map to an area of gene A that has a consensus region with an area of gene B. And so in this case, this purple read is aligning to both gene A and gene B. And so when we do the quantitation, should we count this read as being from gene A or as being from gene B? And then similarly, when we're looking at isoforms, we can have reads that map to different splice isoforms. So for an example here, we have this one gene here that has two different isoforms. And this isoform here includes exon 3, whereas this isoform here does not include exon 3. And we have this read, again in purple, that's mapping to this region of this gene, which is spanning the splice junction between exons 1 and 2. And that splice junction of exon 1 and 2 exists in both isoform A and isoform B. So if we have this purple read that maps to that location of this gene, and that location is present in isoform A and isoform B, which isoform do we think it came from? How do we quantitate that? And so non-weighted quantitation tools will have a read count if it overlaps one nucleotide or more with one gene. And multi-mapped reads and non-weighted quantitation tools will either not be quantitated at all, so they'll just be thrown away and not counted, or it'll be assigned based on an option in the tool. And I should say that the tool option could basically either pick a gene at random to assign it to, or an isoform at random to assign it to. The tool could split the read exactly in half, or however many places it maps to, and exactly proportionate based on how many different locations it maps. So again, that's not weighted, not taking into account anything else. And that'll generate gene quantitation in raw counts. Some examples of these non-weighted quantitation tools include HiSeq Count, and they have 3 different options in this tool, none, all, fraction, or random. So none being if there's a multi-mapped read, it just throws it away and doesn't count it. All means if there's a multi-mapped read, it counts it as a full read mapping to each location. You can use the fraction setting, which will split it up, split that read up in fractions based on how many places it maps. So if it maps to 3 places, it would count 0.333 to each gene that it maps to, or it could randomly assign that read to one of the genes that it multi-maps to. So that's for HTC count. There's feature counts, which have 2 options for multi-mapped reads, which is none or all. So basically it either doesn't count a multi-mapped read at all, or it will count it as one mapped read to each of the locations that it comes from. Like I said, STAR, the tool that we use to do alignment, you can actually add an option to STAR, which will output quantitation. And the quantitation it uses the default of HTC count. When you have STAR give you a quantitation, and the default of HTC count is none, so it throws away multi-mapped reads. There are also weighted quantitation tools. So weighted quantitation tools will use an algorithm known as the maximum likelihood estimation method to assign reads to genes or transcripts. Some examples include Salmon and Callisto. So these are those pseudo-aligners that I was talking about during the alignment. So they use maximum likelihood estimation to determine and to quantitate where reads come from relative to the reference. And I have links here, I should say, for all of these tools if you guys want to learn more about them. Another weighted quantitation tool is the RNAseq by expectation maximization, or RSEM. So this is the tool that we use, and it quantitates uniquely aligned reads, and then it uses the maximum likelihood estimation to calculate abundance estimates to assign that multi-mapped read accordingly to a particular gene or isoform. So I'll go over this in more detail in the next couple slides. And then I should also say here, these output, Salmon Callisto will output transcripts per million and predicted raw counts. RSEM gives you a variety of different quantitation outputs. So it'll output just the raw counts of the templates. It'll output transcripts per million, or it'll calculate fragments per kilobase per million. And these are, again, our links to all these tools if you want to learn more. So counting aligned reads with RSEM. So how RSEM deals with multi-mapping and splice isoform quantitation issue is it uses a maximum likelihood estimation algorithm to essentially rescue reads that are not uniquely mapped by allocating them to one or more transcripts using a three-step algorithm. It'll first estimate abundances based on uniquely mapped reads only. So it'll take a look at all the uniquely mapped reads and try to get an idea of which genes are expressed at which level based on just uniquely mapped reads. And then for each read that maps to multiple locations, it'll divide it between the transcripts to which it maps, proportional to the read abundances estimated in the first step, using this maximum likelihood estimation. So it'll look at how the uniquely mapped reads are mapping to genes, and then use it based on what genes are being expressed according to uniquely mapped reads to then allocate a portion of the multi-mapped read to one or more locations of where it's being mapped. And then it'll go through and compute abundances based on those updated counts, which now includes the fractions of the multi-mapped reads. And so just to do this in picture format, so getting back to our example here, what RSEM will do is it goes ahead in this case, right? So this is going back to that example I had before, but this time what it does is the RSEM package will first take a look at reads that map uniquely, right? So using the example from before, all of these black reads here are going to map to either gene A or gene B, right? So we see here that we have several reads, 8 reads mapping to gene A, or 8 templates, I should say, mapping to gene A. In gene B, we only have one template that maps uniquely to gene B, right? And so now, when we're trying to figure out should the read be assigned to gene A or to gene B, it uses a maximum likelihood estimation to say what's the likelihood that this read came from gene A, knowing what we know about those uniquely mapped templates, and what's the likelihood that this purple read came from gene B, knowing what we know about the uniquely mapped templates to there. So now, given this information, it calculates this maximum likelihood estimation based on these uniquely mapped reads. And in this case, we see that the majority of the uniquely mapped reads were derived from gene A as opposed to gene B. So the maximum likelihood estimation calculation will estimate that this read that's multi-mapped maps to both gene A and gene B more likely came from gene A, just because we're seeing more of gene A being expressed when we look at the uniquely mapped reads compared to gene B, although there is some uniquely mapped reads that we do see from gene B. So then it will split up this read, it'll fraction this read, and it'll give the majority of that fraction will be assigned to gene A, and then a small portion of that fraction will be assigned to gene B. And similarly for different splice isoforms, so again, going back to our example here, this time we're looking at, in black, reads that are uniquely mapped. So this is the first step in the RSEM algorithm. So we see here in order to know if it maps to isoform A or isoform B, we would have to know if exon 3 is present or not. And so you see here these 4 reads that we have in isoform A are uniquely mapping to isoform A, right? Some portion of exon 3 is in all of these reads, so that's how we know that it maps to isoform A uniquely. And then these 4 reads here are mapping to isoform B because we see here that it spans a splice junction between 2 and 4, and no read in between it is mapping to exon 3. So these are all going to be uniquely mapped to isoform B of this gene. So now when we have that read that we looked at previously that could be mapping to either isoform A or isoform B, since they both have exons 1 and 2, and we see that this read maps to exon 1 and exon 2, spanning that splice junction. Now that maximum likelihood estimation method is going to put a likelihood estimate on where this purple read came from, either isoform A or isoform B. And since we have the same number of templates mapping uniquely to isoform A as we do templates mapping uniquely to isoform B, the maximum likelihood estimation will likely assign about half of this read to isoform A and about half of this read to isoform B, because the likelihood that it came from isoform A or isoform B seems to be about 50-50. Before I get into building an RSEM index, questions about RSEM and how it handles multi-mapped reads? Pretty clear? This is why we like RSEM, because it is going to recover those multi-mapped reads, because we don't like to throw away data. And it does it in a way that it looks at all the uniquely mapped reads around it, and then from that information makes more of, if you will, an educated estimation of where that multi-mapped read could have came from. So yeah, this was another thing that was hotly debated with the AWGs, and we settled on using RSEM to do quantitation. Okay, so similar to our STAR tool, as I mentioned, RSEM, in order to run it, does require us to build an index, and that's essentially used to interpret information provided in the BAM file, similar to STAR. The RSEM program requires this index, which is going to be derived from our reference, so our GTF files and our FASTA files for the organism of interest, which in our case is mouse. So RSEM has this part of their tool, or algorithm within their tool, called RSEM prepare reference, which is going to be the function of RSEM that you're going to use to prepare the index for the organism that you're working with. So this tells the RSEM program that we'll extract the reference transcripts from the reference genome using the GTF file and use that to create an RSEM index. And then you have to provide it with a GTF file using this option --GTF. And so we're going to give it our GTF file from Ensemble for mouse, Mus musculus. And then it expects 2 positional arguments to come at the end of the command. The one before the last should be a FASTA file of your reference genome. So again, for us, it's going to be mouse. And then the last part of the command, RSEM expects it to specify the directory for where to output the RSEM index. And similar to STAR, but even better, this only has to be done once, and it doesn't take into, it's not doing alignment, right? It's just counting something that was already aligned in our case. So it doesn't need to be done more than once for an organism, which is great. Once we generate that index, we will then run quantitation with RSEM using these parameters. So I'm going to quickly go over these parameters. Again, all of them can be found, and you don't have to memorize any of these. They can all be found in the RSEM documentation. I have a link to that at the bottom of this slide. And so this time we're going to use, when we actually do the quantitation, we're going to use the RSEM calculate expression function of RSEM, which calls the RSEM program that will estimate the gene and isoform expression from the RNA-seq data. We're then going to use this option ---- alignments to let RSEM know that the input file contains alignments. And then we're going to use this ---- option to let RSEM know that the alignment file that we are providing them is going to be in BAM format. We're also going to let RSEM know with this option ---- paired end that these data to expect paired end, again, because that way they're quantitating the template which is indicative of both the forward and reverse read, right? So quantitating the templates with this paired end option. This seed option, in order to perform the algorithm for the maximum likelihood estimation, it uses a random number generator, and this essentially just needs to be a non-negative 32-bit integer. So we'll just, I think we use 1, 2, 3, 4. 1, 2, 3, 4, 1, 2, 3, 4, 5 for this one. So just a random number generator that's used for the maximum likelihood estimation calculation. And then we have this option here, --seed length. And so this is telling RSEM the read length that we want it to use to count. So the default for RSEM is actually 25 base pair read length. But if you remember from our trimming, and then also in our star, so from our trimming, our lowest read length is 20 base pairs. And then when we do star, we also tell it lowest read length is 20. And so again, for RSEM here, we want to make sure that we tell RSEM that 20 base pair read lengths is our smallest read length that we still want it to accept for the quantitation. This --estimate RSPD tells RSEM to estimate the read start position distribution from the data. So what we'll see is that RSEM will tell us the read lengths of the genes, or gene lengths, I should say, and transcript lengths. And so we're telling RSEM to get the transcript length information from the data. No BAM, --no BAM output. It tells RSEM not to give us back any BAM file. So you can also run different aligners through RSEM. So RSEM can call, star is one of the aligners. The other aligner might actually be BWA. So you can just run RSEM and call star within RSEM to do the alignment in one step. The caveat there is that the aligners that RSEM will run under the hood, the option, it's limited in how you can modify the options for those alignment tools, which is why we do it as 2 separate steps. So this is just telling RSEM, who has the capability of doing alignment if you want it to, that, you know, we don't want you to give us back any BAM files here. We have this --strandedness option, which is important, and why we have to do the infer experiment, because we have to tell RSEM what is the orientation of the reads relative to the reference. So that way it knows to quantitate reads that are, if it's stranded, that are in whatever orientation relative to the reference that we know the strandedness is. So of course, for our case, we just saw that our data are stranded in the antisense direction. And so the option that we would use there is reverse, which basically says that most reads are antisense relative to the reference. And if you're working with paired end data, it would be most forward reads are antisense relative to the reference. So that's the option that we'd be using here. It then expects 3 positional arguments at the end. The third to last one would be a positional argument to specify the input BAM file. So which BAM files to do the quantitation on. The second to last one would be the positional argument to specify the path to the directory where the RSEM reference is stored, and also if you gave it a prefix, what its prefix is. So that's in the slide before this where we built that RSEM reference. We'll have to tell the RSEM quantitation tool where that reference is. And then lastly, it expects to be given a path and also a prefix, if you want, for the output files that RSEM generates. And once we run RSEM, we're going to get a couple of outputs. So the first output, or not the first, one of the outputs that we'll get is an output that ends in genes.results. And in addition to this one, because I'm only going over the genes.results, we'll also get a file that ends in isoforms.results. So the RSEM essentially outputs these 2 files containing expression estimates relative to genes or relative to isoforms. For differential gene expression analysis, we're going to be using the estimates per gene. And so just taking a quick look at that genes.results file format, which it's 9:30 now. So we'll take a look at this genes.results file format and go over it after the break. So go ahead, take your 20-minute break, and please be back here by 9:50 a.m. Pacific time.